Hello everyone, I'm myself Dr. Dipti. I'm MD PhD Physiology and your educator for Physiology. Today we'll discuss about properties of cardiac muscle, that is contractility. Before starting the class, I would like to inform you that you will get soft copies as well as hard copies of all my lectures. To get soft copies, you have to download the application, that is Vesalius, and subscribe. And to get hard copy, you have to order my book, that is Physiology Notes, available on Flipkart as well as Amazon. Also, you can join us on WhatsApp. Our WhatsApp group is there, and number for joining the WhatsApp group, that is 78638 Link for the WhatsApp group, that is also given in the description box. Okay, link for all my book, all my application and WhatsApp group, that is all given in the description box. Now let us start with the properties. We have discussed electrical properties: excitability, automaticity, rhythmicity, and conductivity. And now we'll discuss about mechanical properties, one that is contractility. Okay, now what is this contractility? Contractility, that is the mechanical response of the cardiac muscle, and this begins just after the onset of the electrical activity, that is onset of the depolarization, and it lasts for 1.5 times as long as action potential. Okay, this mechanical response, it overlaps electrical response, because mechanical response that lasts for 300 milliseconds, electrical lasts for 200 milliseconds. And when the adequate stimulus is applied, cardiac muscle, they have ability to generate force and to contract. Now, here you can see sequences of events as we have discussed. Excitation followed by the contraction, that is known as excitation contraction coupling. Calcium ions are required for this excitation contraction coupling. I'm not going in much detail because this we have already discussed. Now, various factors, they affect this excitation and contraction, if contractility of the cardiac muscle. They are: one is all or none law, second is summation of subminimal stimuli, third is staircase effect, fourth one, that is effect of temperature, effect of preload, and effect of afterload. We'll discuss one by one, starting with all or none law. What is all or none law? You can see in this diagram. When the adequate stimulus is applied, heart contracts, and when heart contracts, it contracts to its maximal ability, and this is all response. And if the stimulus is not adequate, there is no response, so that is all or none response. And this is applicable to syncytial arrangement of cardiac muscle. And as we have discussed, heart muscle has two syncytium: functional syncytium, atrial and ventricular syncytium. Atrial and ventricular syncytium. Okay, fine. Now, for demonstration of all or none law, it can be demonstrated on the quiescent heart. How? Suppose on the quiescent heart, or you can say after stimulation of vagus nerve, heart gets stopped, and after that, if you are applying subthreshold stimulus, there is no response. And when you are applying threshold stimulus, you will get highest response. But if you increase the stimulus when it is supramaximal, the response cannot be increased. Okay. Now, second factor that affects the myocardial contractility, that is summation of subminimal stimuli. When subminimal stimuli, they are applied, they get summated. Okay, and this is also studied on the quiescent heart or resting heart. So you can see here, subthreshold stimulus does not produce any response, but when this subthreshold or subminimal stimuli, they are given at interval of half to 1 second, this stimuli, they get summated, and you will get summated response, and this is known as temporal summation, summation of time. You can see here another property, that is staircase phenomena, which is also known as Trappe, that is also observed in quiescent heart. You can see, or after stimulation of vagus nerve when heart has stopped. After stimulation of vagus nerve when heart has stopped, and then heart has started its contraction, initial few contractions, you can see here, they are in the form of staircase. Okay, and that is because of the beneficial effect. Each successive stimulus is benefited by the previous one. Now, what are the causes of beneficial effect? You can see here, when the heart is not contracting, there is increase in the sodium ion concentration inside the heart. Okay, sodium ion concentration increases and potassium ion concentration decreases inside the cardiac muscle cell, with increased calcium influx inside the cardiac muscle cell. So calcium influx when increases, you can see here, when sodium increases, along with that calcium also increases, and when calcium ion concentration increases, it increases force of contraction of the cardiac muscle. So that is the cause for beneficial effect. Okay, Another factor that affects contractility, that is temperature. With increase in the temperature, you can see here, this is normal, this is warm ringer, you can see heart force of contraction also increases and heart rate also increases. Okay, now another factor affects the contractility, that is the effect of preload. Preload means the load acts on the muscle before muscle started contraction, before starting of the contraction. So preload increases initial length of the cardiac muscle, and according to Frank Starling's law, as we all know, what is Frank Starling's law? Within physiological limits, force of contraction is directly proportionate to initial length of muscle fiber. Means more the initial length of muscle fiber, more will be the force of contraction within physiological limits. That this was given by Frank Starling in 1910. Okay, and another important thing, within physiological limit is very important, because if it is more than physiological limits, what happens, larger the initial length, that it results in decrease cross bridge formation. Okay, and that will not increase the force of contraction. So within physiological limit is very important thing here. So Frank Starling's law explained that when large amount of venous return comes to heart, then cardiac output increases. You can see here stroke volume increases when end diastolic volume or pressure increases, that is preload. End diastolic volume means volume of the blood that is present in the heart at the end of diastole, but this is for normal heart. In case of heart failure, if we are trying to explain the Frank Starling law, that is not applicable, because in heart failure heart cannot contract to get the normal amount of blood ejected. Okay, now so that is the limitation of Frank Starling. So you can see here, this is the normal at rest, normal during exercise. Again you can see that end diastolic volume increases, force of contraction increases, but this is not true for heart failure, not true for myocardial depression. Okay, now another important thing is preload. Preload, by how it affects the force of contraction, because of preload there is partial contraction of contractile components. Contractile component of the muscle, they are actin and myosin, and stretching of series elastic component, they are tendons. Okay, so when there is contraction of contractile component and stretching of the series elastic component, that results in absence of the stretching of the muscle as a whole. And so you can say that, you can see, suppose contractile components are contracted but series elastic component are stretched, so you can say that length of the muscle remains same. So this type of contraction is known as isometric contraction. And when we are discussing the length tension relationship, you can see here, this is the relationship between the length of the muscle fiber and the tension. Okay, so you can see here, when length increases, okay, then the tension increases, but here initial length remains same. Okay, this is isometric contraction. Another here is diastolic intraventricular pressure. Here again, and diastolic volume and the tension. Relationship between the diastolic pressure and the tension. Okay, here this is the relationship between diastolic pressure and diastolic volume in the ventricle, and it measures the passive tension. This is your passive tension which is measured. Okay, so as end diastolic volume increases passively, you can see here the tension increases. Okay, so this measures the passive tension. Next curve, that is systolic intraventricular pressure curve. This one systolic. So that is the relationship between systolic pressure and the volume. Okay, so it measures active tension, and that is isometric contraction, known as isometric contraction. Okay, figure shows that with increase in the end diastolic volume, okay, you can see here tension increases. Okay, and that is because of beginning of disruption of myocardial fibers. Now another is effect of afterload. Afterload means load acts to the muscle after muscle starts contraction, and that is the resistance against which heart has to pump. So afterload of the right ventricle, okay, that is pulmonary arterial pressure. You can see here, suppose this is your schematic diagram. This is your left atrium, left ventricle, right atrium, right ventricle. Okay, so right ventricle, the afterload is the pulmonary arterial pressure, and for left ventricle, afterload is aortic pressure. Okay, so when this afterload is increased, you can see heart has to pump more. Always aortic pressure is more. Okay, you can see here that pressure against which heart has to pump. You can see this is the normal effect of end diastolic volume and on the stroke volume. When afterload, you can see decreases, stroke volume increases, and when afterload increases, then stroke volume decreases. Okay. Now, so this I think I am not going in much detail of this one. Uh, Sorry. So now another is effects. Here various effects they were found here. There is complete contraction of contractile components of the muscle and no further stretching of series elastic component. You can see here this one. It produces shortening without developing further tension, and this is your isotonic contraction. Okay, so in one this is isotonic contraction, this one. In B, we have found isometric contraction. What happens here? Contraction of contractile component but stretching of series elastic component. So length remains same. Okay. So muscle contracts with shortening. Contractile components, they are contracted but there is stretching of series elastic component. Okay. And the C, it shows again isotonic contraction in afterload condition. Okay, this one. Contraction of contractile component is there. Okay, but the stretching is not enough and it will not maintain the length. So the contraction is isotonic. Muscle tension increases more than load and that results in isotonic contraction. There is no further stretching of series elastic component that is possible. Okay, another is force velocity relationship. You can see here there is a relationship between the force and velocity of shortening. Okay, relationship between force or load, both the things are same, and velocity of shortening. What is load? That is the resistance against which heart or ventricle has to pump the blood, and velocity, that is equal to stroke output. More the velocity of shortening, more will be the stroke output. Okay, so you can see here as the load increases, velocity of shortening decreases, or you can say stroke output decreases. Okay, now here one important thing is V maximum. You can see here this is V maximum. What is this V maximum? When your load is zero, this is load and tension relationship. When the load is zero, you can see here. Okay, velocity of shortening, that is maximum, this one. And as the load increases, velocity of shortening decreases. This is for normal heart A. And the load at which velocity of shortening, that is zero. Okay, that is P0. Okay, now here in the B, this one, this B shows the effect. Figure B shows effect of stretching when the initial length of the muscle fiber increases. Okay, when initial length of the muscle fiber increases, what happens? V maximum remains same, means the load at which velocity of shortening is maximum, that is same, but P0 increases. You can see here this P0 increases without change in the V maximum. Okay, and when we are comparing this with C, what is C? That is effect of catecholamines. When catecholamines are inserted or when the catecholamines are given, what happens here? There is increase in V maximum also here, and there is also increase in P0. Okay. So what is the significance of force velocity relationship? You can see here after load when increases, when load increases, you can see here stroke output decreases, and end diastolic volume also, and end diastolic volume increases. Initial length of the muscle fiber increases, so force of contraction increases, and the P0 is increased and V maximum is achieved. Okay. Now pressure against which heart is pumping the blood, when the pressure is reduced, initially you can say that heart has to pump against less pressure. So what happens? Stroke output increases, but after some times, this is transient, but after some time stroke output decreases. Okay, and it reaches to the previous level. Okay, now there are effects of various ions, sodium, potassium and calcium, that we will discuss. Sodium ions, when they are low, they produce low voltage ECG. And what is the effect of potassium level? Hyperkalemia, that results in decrease in the resting membrane potential. So that when the resting membrane potential decreases, you can see here this one, that results in unexcitable heart and heart stops in diastole. Okay, and hypokalemia means potassium level when decreases, it results in prolongation of PR interval. Here in the ECG, U wave becomes prominent, this one. ST segment is depressed and T is inverted. These are the effects of hypokalemia. Okay, And what are the effects of increased level of potassium ions? Okay, hypercalcemia. Calcium ion concentration increases. Okay, when calcium ion concentration increases, high level of calcium, heart fails to relax during diastole. So heart stops in systole. Okay, this is normal ECG. This is hypo and hypercalcemia. We will discuss more in detail with the ECG also. In hypocalcemia, you can see prolonged ST segment and prolonged QT interval. This one. Whereas in hypercalcemia, ST segment is shortened and QT interval is also short. Okay. Next property of the heart, that is refractory period. What is refractory period? Is the period during which cardiac muscle does not give response to any stimulus. Okay, normal refractory period of atria, that is 150 milliseconds, and for ventricle, that is 250 to 300 milliseconds. Okay, now what are the advantages of long refractory period? Okay, another, first of all, we will discuss about absolute and relative refractory period. This refractory period has uh, two types. One is absolute and second is relative refractory period. Absolute refractory period, this one, it is about 180 to 200 millisecond. It extends from phase 0, you can see here, in the action potential, to half of the phase 3. This one, phase 0, 1, this is 1, this is 2, and this is 3. Okay, and here, this is very long in cardiac muscle, and it occupies the whole of the systole and initial part of the diastole. This one, this is systole contraction, and this is diastole also. Okay, now you can see here, this is your absolute refractory period, this one, up to this one. And next is relative refractory period, that is about 50 milliseconds. Okay, during relative refractory period, muscle gives response when the strength of stimulus is increased to maximum. Okay, and this period extends from second half of phase 3 here, this one, and phase 4 of the action potential. Okay, and it occupies remaining duration of the diastole in mechanical activity. You can see here, this one. Okay, what is the significance of long refractory period in cardiac muscle? Because contractile process is more than half over during absolute refractory period. Okay, during this absolute refractory period, this contractile process of the heart is completed. So there is no summation. Summation is not possible. So cardiac muscle, they cannot be titanized. So this is protective and this is helpful for proper blood flow to the heart. Okay, and this refractory period is also affected by heart rate. When heart rate increases, refractory period decreases. Also, it is affected by bacterial toxins, various drugs, sympathetic and parasympathetic stimulation also. Okay, so this is about another important period, that is dangerous period. You can see immediately after the end of action potential here. Okay, this is also known as supranormal period. Excitability of cardiac muscle, that is supranormal, just at the end of action potential. During this period, if weak stimulus is applied, then also it can excite the muscle. So this is known as dangerous period. Okay, now experimental demonstration of the refractory period, you can see here. When during diastole, relative refractory period, if we are giving the stimulus stronger than normal normal strength, you will get extra systole. Here you can see, this is extra systole, and which is followed by compensatory pause. Why compensatory pause? Because when normal stimulus comes here, at that time your heart is in absolute refractory period, so heart cannot be stimulated. Okay, so you have extra systole and compensatory pause if you are giving stronger than normal stimulus during diastole period. Okay, significance, as we have already discussed, that because of the long refractory period, heart cannot be titanized and heart cannot be fatigued. Okay, so this is all about properties of cardiac muscle. We are ending here, and again I would like to inform you that you will get all my notes in soft copy as well as hard copy. To get the soft copy, you have to download the application, be serious and subscribe. And for getting hard copy, you can order my book Physiology Notes, that is available on Flipkart as well as Amazon. Link for all soft copy and hard copy, my application and book, they are given in the description box. And you can also join our WhatsApp group. The number is 78638226668. Link for joining the WhatsApp group is also given in the description box. Okay, thank you so much. If you like my video, you can like it, you can share with all your friends, and you can subscribe my channel. Thank you so much.